ओके ओके यस टक प्रोग्रामको लागि आमन्त्रण स्वीकार गरिदिनु भएकोमा यस समाजका आजीवन सदस्य तथा यस विभागका पूर्व विद्यार्थी तथा फ्याकल्टी डाक्टर इन्द्रदेव साहुज्यूलाई धन्यवाद दिन चाहन्छु। साथै उहाँको ब्रिफ सिभी वाचन गरिदिनु हुन प्रोफेसर डाक्टर राजु खनाल सरलाई हार्दिक अनुरोध।
Okay, namaste and good morning. यो टक प्रोग्राममा इन्द्रदेव साहुजी डाक्टर इन्द्रदेव साहुलाई वेलकम गर्न पाउँदा म एकदम खुसी छु र म धेरै टाइम नलिकन ब्रिफली उहाँको सिभी यहाँ अलिकति बायोग्राफी पढ्छु। डाक्टर इन्द्रदेव साहु अहिले मैले नेपालीमै बोलेँ है मलाई ठ्याक्कै प्रोनाउन्स गर्न आउँछ कि आउँदैन ए यहाँ हो मैले सुन्ने ठाउँ। ओके गुड। सो डाक्टर इन्द्रदेव साहु अहिले क्याम्प्सविल युनिभर्सिटी केन्टकीमा एसिस्टेन्ट प्रोफेसर हुनुहुन्छ, फिजिक्सको एसिस्टेन्ट प्रोफेसर र आई एम भेरी प्राउड हि इज वान अफ आवर एल्युमनी। उहाँले 2002मा हाम्रो सेन्ट्रल डिपार्टमेन्ट अफ फिजिक्स त्रिभुवन युनिभर्सिटीबाट फिजिक्समा एमएससी गर्नुभयो र अफकोर्स उहाँको त्यति बेलादेखि नै इन्ट्रेस्ट बायोमेडिकल फिजिक्समा थियो र उहाँले त्यसपछि केही समय यहाँ टिचिङ पनि गर्नुभयो हाम्रै फ्याकल्टीमा 2005सम्म युएस जानुभन्दा अगाडि र उहाँले सुनी अल्बानी न्युयोर्क युएसएबाट 2009मा बायोमेडिकल फिजिक्समा पिएचडी गर्नुभयो र त्यसपछि उहाँले थुप्रै पोस्ट डक रिसर्च एसोसिएटको पोजिसनहरू लिइसकेपछि उहाँ अहिले क्याम्प्सविल युनिभर्सिटी केन्टकी युएसएमा 2019देखि एसिस्टेन्ट प्रोफेसर हुनुहुन्छ र उहाँको अब कुरा गर्दाखेरि अहिले फिजिकल सोसाइटी नेपाल फिजिकल सोसाइटीले अर्गनाइज गरेको हुनाले त्यो पनि सान्दर्भिक हुन्छ। उहाँको थुप्रै मेम्बरसिप छ, जस्तै अमेरिकन फिजिकल सोसाइटी लगायत सबैमा र नेपाल फिजिकल सोसाइटीको उहाँ लाइफ मेम्बर हुनुहुन्छ र उहाँले थुप्रै अनर्स अवार्ड्स र ग्रान्ट्सहरू पाउनु भएको छ। त्यो मध्ये एउटा नोटेबल अहिले रिसेन्ट ग्रान्ट इफ आई एम नट रङ एनएसएफबाट उहाँले एउटा रिसर्च फन्डिङ पाउनु भएको छ र उहाँको अल टुगेदर पेपर बुक च्याप्टर कन्ट्रिब्युसन गरेर 120 जति पब्लिकेसन छ जुन मैले गुगल स्कलरमा रिसर्च गेटमा देखाएको थिएँ। सो आई एम भेरी ह्याप्पी टु रिड brief CV of Dr. Indradev Sahu and I look forward for his talk. Malik sir, please.
Thank you, Raj sir. अब मैले यो फ्लोर चाहिँ इन्द्रदेव साहु सरलाई हस्तान्तरण गरेँ फर प्रेजेन्टेसन। अब कसरी गर्ने स्टप गर्नुपर्छ, स्किप गरेर दिवान नेक्स्ट फेरि त्यहाँ पनि देखियो त सर, यो नदेखिने कसरी गर्ने? Okay. Okay, thank you.
Good morning and namaste everyone. सबैजनालाई नमस्कार। धेरै धेरै धन्यवाद हरिशंकर मल्लिक सर, डाक्टर हरिशंकर मल्लिक सर र डाक्टर राजु सर इन्ट्रोड्युस गर्नुभएकोमा र मेरो ब्रिफ सिभी पढ्नु भएकोमा र मलाई यहाँ आफ्नो रिसर्च बारेमा केही जानकारी दिनको लागि जुन मौका दिनुभयो त्यसको लागि नेपाल फिजिकल सोसाइटी र सेन्ट्रल डिपार्टमेन्ट अफ फिजिक्सलाई धेरै धेरै धन्यवाद। र आज म यहाँहरु समक्ष केही आफ्नो कामहरु बारेमा केही स्टोरीहरु शेयर गर्न चाहन्छु र टाइटल आज मैले एक्सप्लेन गर्ने प्रोग्रेस इन बायोफिजिकल टेक्निक्स फर स्टडिङ मेम्ब्रेन प्रोटिन। यहाँ मैले यहाँ स्लाइडमा एउटा इमेज राखेको छु। यो इमेज यसरी हेर्दाखेरि यो एनर्जी लेभल हो। यहाँ एउटा म्याग्नेटलाई देखाइएको छ र जब म्याग्नेटलाई म्याग्नेटिक फिल्डमा राखिन्छ अनि यसको चाहिँ दुईटा स्टेट हुन्छ, स्पिन अप र स्पिन डाउन। यो चाहिँ जस्ट एउटा एनिमेसन हो र यतातिर चाहिँ इलेक्ट्रोन प्यारामाइट रेजोनेन्सको डिफ्रेन्ट चाहिँ मेथडबाट कुनै पनि सिस्टममा प्रोटिनमा भएको डाटाहरूलाई देखाइएको छ र एउटा जस्ट इक्जाम्पलको लागि देखाइएको छ। र आज म यो टकमा प्रोग्रेस इन बायोफिजिकल टेक्निक्स फर स्टडिङ मेम्ब्रेन प्रोटिनमा अहिलेसम्मको फिल्डमा के कस्तो प्रोग्रेस भइरहेछ र हाम्रो कन्ट्रिब्युसन के छ त्यसमा, त्यसको बारेमा हामी भन्न चाहन्छु। र यहाँ सुरु गर्नुभन्दा पहिला चाहिँ म अलिकति हालत भन्न चाहन्छु। अहिले यहाँ कुरो गर्नु यहाँ रिसर्च सम्बन्धमा च्यालेन्जेस एन्ड अपर्च्युनिटी इन मेम्ब्रेन प्रोटिन रिसर्च। मेम्ब्रेन प्रोटिन रिसर्च हामीले किन गर्छौँ र यसमा आउने च्यालेन्जेस र अपर्च्युनिटी कस्तो छ त्यसमा हामी कुरो गर्छौँ। अनि डिफ्रेन्ट बायोफिजिकल टेक्निक्स जुन अहिलेसम्म फिल्डमा छ त्यसलाई चाहिँ मेम्ब्रेन प्रोटिन स्टडी गर्नको लागि कसरी एप्लाई गरिरहेको छ त त्यसको बारेमा हामी डिस्क्राइब गर्छौँ। र जस्ट ब्रिफ इपिआर स्पेक्ट्रोस्कोपीको बारेमा बेसिक एप्रोच चाहिँ यहाँ डिस्क्राइब गर्छौँ। र सम इक्जाम्पल्स फ्रम माई रिसर्च एन्ड रिसेन्ट पब्लिकेसन्स just to describe the how we can solve the biophysical problems or any biomolecules or any other things using electron paramagnetic resonance technique. And finally, future perspective and summary I will describe. So let's start with why we study membrane protein. So before going through that, we have to understand what is membrane protein. So membrane protein means anything that is associated with the membrane. Membrane proteins are included in several life processes. They are very important for the living organism. For example, here we can explain signaling, transport, recognition, and catalysis. Based on the recent information, we have more than half of the modern medical drugs. They are related to the membrane proteins. So membrane proteins are very important. Also, in the right side, you can see membrane proteins. They expand the bilayer in different ways. Sometimes there is single transmembrane section, multiple transmembrane sections. Sometimes it interacts with the surface and some other protein as well. So overall, if we see in several examples, membrane protein just acts like a communication between the inner and outer part of the cell. So membrane proteins are abundant and important. Let's go through the research area of the membrane protein. What are the current research area? When we talk about the membrane protein, what we need to know? So in this case, here we see structure, dynamics, and environment. In order to understand the function of any biological system, any macromolecule, we have to understand how they have the they are organized, they are arranged, and how their dynamics is, how they are moving. In order to understand, when we combine the structure and dynamics, then we can say what is the function of that system. But in the case of membrane protein, we have to also focus on the environment, because environment surrounding is very important. When membrane protein is embedded, any type of the membrane system or any kind of things, we have to understand how it is interacting. So especially for the membrane protein, environment is very important. Here I am going to talk a little bit about the what kind of the membrane environments are currently available in the field and what we have done to improve that. Okay, so most common is the micelle. Micelle has the single acyl chain, and it is smaller. It doesn't have lipid barrier, so it is very poor membrane mimetics. Membrane mimetic means it provides the soluble environment, soluble environment, or like a host for that particular protein. Next is vesicles. Vesicles is spherical. It has the lipid barrier, but it is very heterogeneous in the nature. So it is challenging. When we incorporate the protein into the membrane environment, it is very challenging to get the proper information about that particular protein in the vesicle system. However, we have the recent artificial bilayer we call the bicell. In the bicell, it is a smaller shape, but it can provide the lipid barrier. But in order to form the bicell, we have to use combination of the lipid and micelle or detergent. And for some cases, some kind of the proteins, it is very challenging to have a specific type of the lipid and detergent system to give the function. So it is still challenging. However, recently people have derived nanodisk. So what nanodisk does, it takes the lipid bilayer, a small portion, and wraps with the scaffold protein. That makes the size smaller. If size is smaller, it is homogeneous and transparent. That is easier. But in this case also, since it is a scaffold protein, if we have the protein inside the system, there may be chance that they can interact. If they can interact, they can disturb their function. So still it is challenging. That means new membrane mimetics is still desirable. We have to find out the new membrane mimetics. And this is one cartoon that explains very well. When we study the membrane protein system, most of time people say it is very challenging to study. But reality is we don't know how to work with the protein system. Let's go through here. Uh, this is the new system that we have developed in our lab. We use the polymer. Instead of using the protein system, we use polymer to wrap the lipid bilayer. So if you see in this image, we have the protein embedded inside, and outside we have a small malic acid and esteric. They provide the environment that doesn't disturb with the protein system, but it can provide the membrane mimetics that is functional, that is close to the native environment. So there is no interference with the target proteins. It maintains biologically relevant environment. If you see in this image, this is just kind of vesicles, because vesicle size is always very big. But when we incorporate into the malic acid and styrene copolymer, we also call lipodistine nanoparticles, it becomes very homogeneous. This is the TEM transmission electron microscopic image. Here we can see it is very heterogeneous, but we have like a homogeneous. Also, if we just take the direct image, it is milky, but when we have a protein into the polymer, then you can see it is transparent. So this is very suitable. We have developed in our lab, and this is very helpful for the using different biophysical techniques for studying membrane protein. Now, when we say membrane protein, it is always challenging to study. What is the status currently in the field? If we look on the protein data bank, based on the protein data bank, total protein structures, including soluble protein or any other things, like a globular protein, including overall, it looks like up to 8, we have 187,844. But if we just look on the membrane protein, we have only 1464. If you see in this trend, this is the membrane protein trend is going on. Overall, it looks like the structure is only 1%. This means it is still challenging, but if we can work a little bit more, we can get more information, more data to report more structures. Although we know that there are 50%, over 50% membrane proteins, they are related to the drug target, but information is not available in the field. So, but although we see here most of the 1% structure for the membrane proteins, they are determined in the mycelium environment, which is not suitable, or which is not reflecting, which cannot reflect exactly the native environment. So this means we need further investigation. So that slide highlights the need for the further investigation on the membrane protein. So that provides great opportunity for working on the membrane protein systems. Okay, now let's look for the what are the biophysical techniques available. They can be used to study this kind of complicated system. Most popular is X-ray crystallography. This is very popular and it can provide very high resolution data, but it has also several limitations. Overall, 89% of current structures they were determined from the X-ray crystallography. Another technique is NMR, which is also popular, but it is also a little challenging. 10% of the current structure is done with the NMR, and recently there is a cryo-EM. It is very latest technique and very popular, but very expensive technique. And sample preparation is always challenging. Overall, when we talk about the membrane protein system, several challenges happen. Hydrophobicity. We know that membrane protein is hydrophobic. It is very difficult to express. Expression yield is very small. Similarly, when we talk about the X-ray crystallography, we have to make a crystal. It is very challenging to crystallize the membrane protein system. However, for the soluble protein or detergent soluble protein, it is easier. Membrane protein is challenging. And it is also very challenging to study in different kind of the lipid bilayer. So we have to go further, higher level of the techniques that can be suitable for overcoming all these limitations and that can provide the additional information. So for this one, I'm going to talk here the new things, two things. One is electron parameter resonance technique, although it was developed very early, 1947 or something like that, but the usefulness or in the biological system, recently it has been developed. So currently we have used that one. From the experimental side, it doesn't have any protein size limitation or anything. Another is any kind of lipid bilayer environment can be used, very sensitive. If we talk about NMR, we use proton. For the electron parameter resonance, we use electron. Electron is 1,000 times lighter than the proton, so it is 1,000 times sensitive, so it is easier to work. Next thing is computational technique. Computational technique is very popular recently. Why it is popular? Because it doesn't require any laboratory, just computer, so it's a little easier. It is also very useful to validate the experimental, because experiment and theory or computational, all of them, they are complementary to each other. So less expensive, easier to work. It is very popular to describe the dynamic function. So if we just combine experimental approach and computational approach, both can provide detailed picture. Biological systems are still very complex and very difficult to study. So interdisciplinary or multidisciplinary technique is very helpful. All right, so let's talk a little bit about the electron parametric resonance technique, because my work is mostly related to this part. So electron parametric resonance, obviously it is a resonance technique that measures the absorption of electromagnetic radiation, especially microwave, by the unpaired electron when we keep that unpaired electron in external magnetic field. This is very well known formula, everybody knows that H nu, Planck's constant, frequency, Landau factor, Bohr magnetron, and magnetic field. This is basic equation for the electron parameter resonance. Since we need unpaired electron, most of time biological systems are neutral. We have to add some kind of tag. So we use nitroxide spin level, which has nuclear spin of 1. So when we go through the multiplicity, it is 3. So it has 3 lines. There is 3 kind of transition. Each transition represents its line. So this is a hyperfine interaction. When we include that one, we get this kind of spectrum for unpaired electron. So this is very useful. We will talk about the different EPR techniques. One is called continuous EPR and another is pulsed EPR. Continuous EPR will give the line shape analysis mostly for the dynamics. Also, it will help to understand accessibility with the environment. Next technique is like a electron-electron resonance, double electron resonance. That gives the distance. It is very easier to get a distance from 20 to 80 angstrom. That is very challenging from any other technique. So it is EPR can give the information about the conformation of the biological system. Here is the, since the biological system doesn't have its own unpaired electron system, we have to add additional functional group to do that. For that one, we use side-directed spin leveling technique. In this case, what we do, we replace the particular amino acid from the protein with cysteine, and cysteine has sulfur. So it forms disulfide bridge and ultimately giving a stable unpaired electron system on the protein. How this works? If we look on this image, this is for the free spin level. If you have just free spin level, MTSL, you will get very sharp. But if it is attached to the peptide, means a small protein system, line shape is getting different. It is getting broader. If it is connected to the bigger protein side, then it gets further like a broader. And for the like a rigid limit pattern, means when it is frozen condition, we get the powder pattern spectrum. So overall, what we see from here, as the heavy molecules or bigger complex is getting, we see the line shape is getting broader. That is very unique information that we can get from the EPR. So for the application, there are several applications. We get the dynamics, side chain dynamics, also backbone fluctuation of the protein system, protein topology. Topology means how protein interacts with the lipid bilayer. That we'll get through, and conformational dynamics. Every biological system is dynamic. There is nothing like a stationary. So we see how it is moving or how conformation is changing. If we understand the conformation, we can understand what is the function of that system. We also get the distance reaction. As I mentioned, for the pulse experiment, we get the distance between the two sides of the protein, and that we can use to get the structure. Also, how protein interacting, we can get from the EPR. So I'm going to know, we already discussed the EPR technique, biophysical technique challenges. Now we are going to give some example of the biological problems that we solve using EPR. So I'm just discuss a little bit about the potassium ion channel proteins. This is very important protein. We can see here when we talk about the Kc and Q1, they are channel protein. So when they interact with the KCNE1, it has several families, E1, E2, E3, E4. So when it interacts, it forms the channel. Channel means flow of the ions. It helps to have the flow of the ions. It regulates the flow of the potassium ion through the potassium channel. Also, the mutations on any kind of this system, either Q1 or KCNQ1 or KCNE1 or any kind of the system, they are linked to the several disease, like a first recent is heart disease, we call long QT syndrome. So if we look on this image, I think most of us are familiar with the electrocardiograph. It has PQRST wave. But when anybody who is suffered from the long QT syndrome, it has the QT interval. This is prolongated. Prolongated means interval is getting bigger. So when the heart function try to revert back, depolarization state, in this case, this is the longer time period and heart cannot come back to the initial state during the contraction and expansion. So in this case, sometime when athletes are doing the exercise or playing the sport, sometime you might have heard some players, they die suddenly on the field. So that is the very challenging part. 1 out of 7,000 people in the world, they are suffered from the long QT syndrome. So this is very challenging system and very useful for the study if you see the heart condition. If anybody has long QT syndrome, sometimes it completely either stops or it delays the function. All right, so I wanted to show how we use EPR to solve this problem. We have KCN1, this is the protein that I discussed related to the potassium ion channel. Its structure is very challenging. It has not determined in the lipid bilayer system. Our collaborators, they did a structure in the micelle using NMR. But since we know that micelle is not suitable environment, we have to study in the lipid bilayer environment. NMR cannot study in the lipid bilayer environment because due to the bigger complex and NMR has the size restriction. When we have the lipid, it becomes bigger and NMR cannot provide the sharp peak in their data because it becomes broader. So we have to go through and since study was only done in micelle, we have to understand how it functions, what are its structure in like a lipid bilayer environment. So there are two reasonable questions coming up. How does KCNE1 interact with the lipid bilayer? Next thing is we have the since it is transmembrane protein, its transmembrane system, it is incorporated into the lipid bilayer. How it is interacting, how it is its conformation, whether it is straight or curved, that helps to understand its function. So two problems we had. And in order to solve this problem, we started with the EPR. How does KCNE1 interact with the lipid bilayer? So in order to go through that problem, we combine the technique like a molecular biology technique and electron parameter resonance technique. Molecular biology technique means we just use the biochemistry approach, microbiology approach to do the cloning and do the side-directed mutagenesis, do the protein expression, prepare final protein. So we start from here like a growing culture. Maybe biochemistry people will know more about that. And then we express the protein from the inclusion, but these are the steps. And we put the side-directed spin labeling condition. Finally, we have the spin label tag on the protein. We incorporate into the micelle, and then we have some protocol we use in the vesicle, and then go through the EPR. When we have the EPR data, we use simulation as well, using the computational technique to theoretically draw the structure and then overlap with the experimental data and draw the dynamic parameters. This is the gel that gives the how pure the protein system is. This is molecular biology. So here are some of the data. As we can see, these are the sites. This is the amino acid sequence. All the red sites are just the mutated site. We studied those sites because for each site we have to do the mutation at a time, and then we have to do the EPR. So if you look on this data here in the transmembrane domain section, this is the transmembrane domain section of the protein. If we choose some of the side like a 50 and 66, we can see the line shape is very different from the side. It is broader. That suggests us when we have a spin level within the transmembrane domain section, its motion is restricted compared to the outside. If we see the outside here, 12 around somewhere around this region, and here 78 around somewhere here, it is broader. So this is the some example I put, but we obtained lot of the data. Almost out of the 129 amino acid, we did around 60 or something like that samples. And this is the side chain mobility data, and this is the delta H. Delta H means inverse central line. Here, if we see here, so this is called delta S, this is called the mobility parameter. We just measure the width of the central line in the spectrum. And when we plot the spectrum, we plot this data for all the residue question that we did. What we found, we saw the significant pattern. The sites that we studied within the transmembrane section, we found that inverse central line with it is smaller in this region. That suggests that these sites are slower because it is embedded into the lipid barrier. But when we go through the N-terminus side, means N-terminus is this side, C-terminus is here, we have higher little bit, but C-terminus is even more high. That suggests that this is more mobile compared to this. These data we got, and again we quantitatively also measure motional parameter we call also rotational correlation time. Rotational correlation time means how the spin level fluctuates within the certain period of time, unit time. When it makes 1 radian, when it rotates, that time we measure. So when we measure, it looks like the tumbling pattern for the transmembrane section is longer. It takes longer time to tumble when we compare from the outside. And additionally, what we did, again, we measured, it is called depth parameter. Membrane depth parameter, again, we use EPR and we measure the distance from center of this lipid bilayer to the different side. When we measure those things, what we saw, that tell us the topology. If we see here 47, 45 to 71, these sides, they are interacting with the lipid bilayer and they're spanning the lipid bilayer. Any other technique is very challenging to get those information. EPR is the only technique that can provide this information. So we determine the topology, how they are going up, and then we summarize based on our data. What we found, we also found that if we, I forgot to mention one more thing here, if you see these small peaks, that means multiple conformation. When protein interact with the lipid bilayer, some population is interacting with the higher motion compared to others. Those kind of information we also get from the EPR. So here we summarize. KCNE1 undergoes multiple conformation, and then we also found the extracellular outside region is more mobile compared to the inside region. We also determine the topology exactly which site is staying at the surface, which site is at the center. Finally, we also found that N and C terminals, they are interacting with the surface, also interacting with the solvent, with different populations. So these are conclusions. Next question was whether this texture is curved or straight. For that one, again we use the EPR, but we use different technique. For this one, we use double electron-neutron resonance spectroscopy. So for double means we put a spin level at two sides, not one side, and then we measure the distances by using one of the double electron resonance. This is the data that we have, and when we do some analysis, we get the distance distribution. So this means this distance we can obtain between two spin levels. So for that one, before going through that, I also wanted to talk a little bit about how DEAR measurement works. This is the Hamiltonian, rotating frame Hamiltonian. Since there are two sets of spins involved, We have frequency of the first spin, frequency of second spin. This is the coupling frequency that we are interested in. How close they are, that will be related to the coupling frequency. If they are very far, that value will be different. This coupling frequency is related to the inverse of the distance, third power of the inverse of the distance. And with certain angle, if we look on that one, here, this is the one spin, another spin, it is the magnetic field, this is the angle between them, the vector, they are connecting, and whatever angle it makes with the magnetic field. Based on data, we collect coupling frequency, and then we obtain the distance by using the formula. This is the 4-pulse gear sequence where we have the different pulse, pi by 2 pulse, 5 pulse. Again, second 5 pulse, there we get the echo. When we apply another frequency at the second set of spin level, it reverts back. So, what is the effect of the second pulse on the this kind of the echo? That will tell us how strong they are interacting, means how far they are or how close they are. This is the theoretical concept little bit I wanted to mention. Here are the some of the data. If you look on those data, what we see here, first side, this is the micelle. This is the lipid bilayer, and this is the lipodisc nanoparticles that I had already mentioned. Lipodisc nanoparticles provided better distance. It means sharper distance. Means if we have sharper distance, this means we can say more accuracy for the distance. This is the single spin level, but if we have double spin level, that can give us even sharper. So these are the some methodological development that we develop, how the technique can be improved to get the better experimental data. All right, so in order to get the confirmation whether it is state or curve, we collected the distance between several stars. Here you can see we have 9 single spin level and 5 double spin level. We collected the data on all these 3 environments, and then we use those data to do this like a computational. We did a computational simulated annealing molecular dynamics simulation, and then we determine the 3-dimensional structure with those sections. So earlier people just used NMR or X-ray crystallography to determine the 3-dimensional structure, but here we use EPR data to refine the structure and get the 3-dimensional structure. So if you see in this case here, this is the overlap 8 structure that we get. We usually calculate 100 structures, keep 8 or 10 structures with the lowest energy. And we compared with the NMR data, what we saw, this is the blue one was the NMR structure that they had earlier, and we got with the lipid bilayer data. We found almost similar conformation, but in the case of the lipid bilayer, the degree of the curvature is a little bit less that we found. So these are the additional information that we got. So we solved these 2 problems, and we found that we can use EPR to solve this kind of challenging problem. There is another problem I wanted to mention. Here is just movie little bit. I wanted to show how this movie works. So this is like a molecular dynamics simulation movie, and what you see here, how the different portion of this protein is interacting with the lipid bilayer. Why it stopped? Why it looks like it is getting slower? Okay. Okay. I don't know. There is some technical things probably. Okay. Now it looks like it. So what we see, this portion, they are interacted with solvent. They are very flexible compared to the inside. We also see sometimes this portion try to come to the surface. They wanted to interact with the surface area. So overall, based on the experimental data and computational data, we can get the clear picture how this protein functions when it is interacting with the lipid bilayer. Means how its structure and dynamics are. Based on that, we can just describe how it can function. Okay, here is another example. I will not take much time here, but just wanted to show another example. This is called bacteriophage Hollin system. They are like a molecular clock. They are responsible for, they have several applications in biotech and pharmaceutical field. They also create a hole and ions can propagate. So we wanted to know what kind of the conformation it has. This is the same system, but in 2 forms. One is having the 2 helix within the lipid bilayer, but in another form, 1 helix is interacting with the surface. So it is very challenging to know how things are coming out. For that one, we applied the EPR again and measure the distance among several places like that. And then we use those distances to again do the three-dimensional structure. And we finally got the structure like that. So we found that these two kind of the conformation of the system, one is within the lipid bilayer for both the helix, and another one is interacting with the surface, and one is inside the lipid bilayer with EPR. Here is another system. Currently, I am working in my lab. This is a different family of the KCNE1 system. This is called KCNE3, similar, but it has 103 amino acids. Its function is little bit different that we had earlier. It holds, it opens the channel. It regulates the homeostasis in the cell, but its mutation again related to the some long-QT syndrome disease, secretory diarrhea, and cystic fibrosis. Again, it is very significant again system, and currently I'm working in the lab. And for this one again, we didn't have the structure in lipid bilayer because NMR people do only in the micelle. They have recently tried to do in the isotropic bicelle, but still they use a lot of the micelle content. So their structures are not reliable. So we tried to again refine this structure. We wanted to use the EPR data and try to solve this problem. So for those one, we again, here are the some of the data we have that I wanted to show. Again, we selected some of the sites and we compare the line shape. This is in detergent micelle, this is in lipid bilayer environment. If you see data from here to here, they are broader compared to here. Again, when we plotted the delta H inverse, this is the inverse central line width. Again, for the micelle, it is bigger compared to the lipid bilayer. Again, lipid bilayer gives the broader spectrum. It restricts the motion. So similar kind of effect we see here. When we calculate the rotational correlation time, again we see that longer for the transmembrane section, but shorter for the outside region. This is not the whole structure. We just try to go through the couple of the data set, and recently we published this paper. I think this is still it is in the press. It accepted, but I think it will come out soon. And now I wanted to show some small clips of the molecular dynamic simulation that I used for this system. So for this case, again, we know that APR, even we do experiment. Experiment is very challenging to study the protein in different environments because you have to prepare the sample, go from one lipid bilayer to another lipid bilayer, very expensive, very time taking. So what we try to do is we wanted to just use the molecular dynamic simulation in all these three environments and compare that. Here is some of the starting phase we use, the protocol. We use NAMD. I think most of you already know the nanoscale molecular dynamic simulation that is free academically. We use the POPC, we use the force field CHARM 36, especially for the surface area for the pre-equilibrated system. This is the initial starting system that we prepare. And then we do the simulation. For this case, where we combine protein plus lipid bilayer, system becomes very big. If we see here, 114,439 atoms. So simulation time will be a little bit longer. This is 105 nanosecond we did initially. And these are some snapshots in different environments, POPC alone, and DMPC alone. And you can see at particular nanosecond time period, the conformation of the system is different. That suggests us protein behaves differently in different environments at particular time frame. That we saw from here. We did also another analysis here. We also wanted to calculate the G distance, means the distance of particular site from the center of the lipid bilayer, and try to see how these data are compared in three different environments. What we saw, the size of the width of the lipid bilayer, POPC, POPC is little bigger size. If we see here, earlier when I saw here, POPC, the size chain is bigger. It is 16 to 18. POPC is 16 again, but DMPC is small, 14, 12 to 14. So in those data sets, we have, based on the width of the lipid, the size of the transparent domain, it changes slightly. That we found. So these are the, again, this is the molecular dynamic simulation data that we have. For the KCN3, we saw the behavior of the interaction of the different sites is slightly different compared to here. Uh, if you see, it has slight small helix here, but this region is mostly flexible here. This portion is sometimes interacting again with the surface and moving outside. So from that one, again, uh, this paper is already published. It is in membrane. We just published maybe in last month. Okay, so I just wanted to now summarize. We can, I gave some of the example. I didn't show most of the work because there is also time limitation probably. So I try to show a couple of example with some of the research, and then try to see how the different biophysical technique can be applied to solve the biological problem. We have found bifunctional spin level that I earlier saw. When we have the two systems together, it restricts the motion more. So we get the better data quality in the distance measurement. Similarly, lipotic nanoparticles that I already earlier saw gives the better mimic system. And we studied KCNE1, KCNE3, pinhole systems to understand the topology and their conformation. We also determined the three-dimensional structure of the both systems that I earlier discussed. And when we use molecular dynamics simulation, obviously it complements the data. Especially, we cannot do each and every point with the experiment. This is very costly, very expensive, very time consuming. But when we get some of the experimental data and we use those as a reference, and then we do the simulation, that gives the better information that we found. So these are the information. Uh, and we try to study the interaction of the protein system in different lipid bilayer environment. Most of time, experimental people do just only one biological system or only one mimetic environment. But if we do the computational work, we can do all. So it is definitely easier, but at the same time, when we only use the computational, again there is again the question. When we use the starting point or like that, whether it is reliable or not. So it is very important to have both experimental and computational. So when we combine, then it is easier to understand. So here are some of the my lab members, and we have several collaborations going on, different works, and there are several funding agencies, they support our work. Recently, National Science Foundation grant is currently going on. Kentucky Academy of Science, they have also supported an affiliate college association. They are the local state level grants, so they are supporting my work, and I would like to thank all of the collaborators, and especially Central Department of Physics and Nepal Physical Society and all of the audiences. Thank you so much, and giving me this opportunity, and I'll be happy to answer any questions.
There are some people in online.
There is someone raised hand.
I want to know what is that? Is the cryo-EM?
Cryo.
Means very low temperature.
Yes.
IEM is automatic.
Yes, cryo just as electron, they use electron definitely. Cryo means very low temperature they use, like helium temperature, or people also use nitrogen temperature as well, liquid nitrogen, 77 Kelvin. For more, some of the system they have to go to lower temperature, and they use the electron beam to get the structure. So for that one, this is very new technique, very expensive technique, but most accurate technique. For those techniques, we don't need much sample. Just one sample is enough to get the structure. They get the high resolution image. This is the latest one. So they can give the three-dimensional structure. But at the same time, it has also limitation. They cannot give the structure for the small system. For that technique, we have to have a bigger one. So definitely there are a lot of the things, but when we combine several techniques, that gives the better information about the system.
In your measurement system, that is EPR, you need the crystals.
No, for me, we don't need crystal.
Oh, you don't need.
No, for X-ray only people need crystal. For me, we need just a spin level. We have to put a spin level. Like a MTSL we call, or any other. Sometimes if you have metal system in the biological, like a metal proteins, metalloproteins or anything, if they have, then we can use that technique, that as well. But most of time they are neutral, so we have to incorporate the
external.
Only thing you need to incorporate that is cysteine or something.
Cysteine, we have to do the mutation because cysteine has sulfur, and we have SPN level, it has also sulfur. So they form like a disulfide bonding, and that is more stable.
That makes the protein stable.
Yeah.
And you use the, your sample can be the liquid.
Yeah, liquid, definitely, liquid. But for the distance, we still use the frozen sample for the distance because relaxation time at room temperature liquid form is very short. So when we want to collect the data, echo dies very fast. We cannot collect all the interactions. So we try to freeze so that relaxation, T2 relaxation gets a little longer, so we can collect all the interaction population. Yeah. But membrane proteins are
How difficult to prepare your sample?
Yeah, it is challenging, but we have the protocol, we have optimized the protocol. Most of the time it takes 1 or 2 years to optimize the system. 1 or 2 years.
3 years.
2 years, yeah, 2 to 3 years. To system, we optimize the protocol. Once we optimize the protocol, then it works pretty well. But it is very challenging because
I don't understand what is that protocol mean. Is it government that
no,
protocol?
we develop. So we develop, for example, we have to do the mutation.
Okay, that is
Yeah. So once we do the mutation, we have to make sure that mutation happens only at our site specific desired position.
that is the.
Yeah, that is also challenging. Once we solve that challenge, then we have to do the expression. Then we have to incorporate protein in the host bacterial cell. Then we have to do the grow the culture. Then we have to go through the yield, means how much protein we can get from the particular liter of the culture.
That is the thing I'm asking. What is the percentage of getting the wanted result from the big sample?
Yeah, so for example, when we grow the culture, like for my system, especially KCNE3 system, right now we are doing E1. So from 1 liter culture, we get around 1 milligram protein or something like that. Still, it is challenging to work, but students are working always 24/7. It is going on in the lab. So people work and then they get the data.
Maybe some student may be frustrated.
Yeah, definitely. Yeah, definitely it is frustrated. Yeah, so okay, go ahead.
Yeah. So the time frame that you mentioned about 1 to 2 years, it is basically the experimental process that you are mentioning.
Yeah, so most of time 1 or 2 year means if things will work, then just in 1 month you can prepare the sample. But in the very beginning stage, when we start with any new system like KCNE3 or any system, when we start, then we have to optimize each and every stage. For example, when we choose the vector, we don't know which vector means will work for that particular DNA. We have to try a variety of the system, and then we choose one that gives best one. Then we start with the expression. Now when we grow the culture, there are several processes, several ways. How much temperature we have to use? We use 37 degree or room temperature. We have to scan several things. Then we decide, oh, particular age, particular time period, particular temperature will give the best yield. That, and then when we go from there, then we have to purify the protein because we just collect the bacteria, and from the bacteria we have to harvest, and then we have to purify from that. So now we have to also optimize how we purify the system. So overall, it takes little while. Yeah, that's why time period is about 1 to 2 years. But once it is optimized, then most of the time students get all the publication within third, fourth year of their PhD, not for first, second year. They spend most of the time. But after around third, fourth, fifth year, they get several papers because once it is done.
It means when you got the right sample, you do the party there.
Yeah, that is it. Most of the time, about 90% that are just wasted.
There is few questions in chat.
Yeah.
I would like to read it from Ambadatta Panta.
Okay.
Slide 21. I have a question. What about temperature dependent behavior? Oh yeah. Go ahead.
Okay. I will minimize 21. Just 21.
Look back.
Oh, I see. Okay. Don't have here.
What about temperature dependent behavior?
Behavior. Okay, so temperature dependent behavior means when we increase the temperature, mobility. For example, from room temperature to like 380, okay, why is coming? 380, 325 Kelvin, we see the sharp pattern. The lines.
Sorry, this data is at which temperature in the sir?
Room temperature.
Room temperature.
Yes.
Okay. Did you check around the freezing temperature?
Room temperature, lines speed data, zero data, and the room temperature. But for the distances one, we use 80 Kelvin. Right here. Let me go here. Okay, these data are at 80 Kelvin. Means liquid nitrogen gas.
Yes. I have one question related to this one also. You are
Okay.
at your peak position. I know with distance, these peaks or how to say frequencies are changes with distance. So what is the field distribution at peak position? I mean, how many gauss or milligauss, microgauss or something? Because this is because of the field distribution.
Yeah, so we use from this is the time, and then it is intensity, and we have used slightly different way of analysis. We use like a take pattern frequency. From the frequency we use L curve we call, and then we get those data. So it is like a around 2 to 4 angstrom that we can say distribution like that. Again, it depends upon the whom we have maximum. We don't go exactly the gauss, but we measure in the like a distance here, like a distance. So, for example, if we have the central peak around 32 angstrom, then distribution we usually say 2 to 4 angstrom, depending upon the different type of the sample. For example, here right side you can see it is very sharp, maybe 1 angstrom with distribution, but here it may be more. So that way we analyze, not exactly, or there is also another way, we can directly use the Gaussian analysis. But here in this case, I have not used Gaussian analysis.
Okay,
We
thank
use
you.
the
Then
Tikhonov regulation.
is this also room temperature?
No, this is at 80 Kelvin.
80 Kelvin.
Yeah. The reason
But
behind that
hydration.
Okay, go ahead. Go ahead.
Please. Hydration behavior, I mean 80 Kelvin and room temperature, role of water is quite different. So how you consider hydration dependent? Your sample is a dry or a hydrated sample? Could you explain?
Yeah, our sample is in membrane. Okay, so membrane bilayer is slightly, its behavior is different than the room temperature. So we use cryoprotectant. Yeah. Your question is when we treat at room temperature liquid one, when we treat, then how it behaves. Definitely, when we freeze, if there is liquid, just water, then it will break the sample tube. We use quartz normally. We use cryoprotectant like a glycerol, or that help us to stabilize the sample for this case. Yeah.
Okay, thank you very much for your answer or reply. It's nice talk. Okay, thank you.
How is your the 80 Kelvin nitrogen temperature is the 77 Kelvin, and you use closed cycle kind of.
So we have like a cryo system, cryostat system. So we feed the liquid nitrogen across the sample region, and it is continuously going on.
That is a closed cycle.
Yeah, closed cycle, that's true. And then we use like a temperature control, there is heater as well. If we have to specify particular temperature, we tune the heater as well as the flow rate. So that way we control. Sometimes we have to use liquid helium to go through 4 degree or 4 Kelvin or 5
Or the
Kelvin.
low temperature.
Yeah, very low temperature. Some of the systems, they have very short T2 relaxation time. For those systems, we have to go to the low temperature. Yeah. Go to
Just I have to check. Yeah. Okay, there is one question from Susil Pangeni.
Okay.
What is the reliability of lipid bilayer you are using? Generally, thickness of bilayer Z value is 4 nanometer, but I see 8 nanometer is one graph. Could you justify?
So yeah, actually, when you say 4 nanometer, this is 40 angstrom. It depends where you measure. So most of time, POPC. If it is POPC, let me go back here.
Okay.
Okay. So here I have here all the information. Okay. So if you see here, if we only use the acyl chain region, only this region, for the POPC it is 36 angstrom. For POPG it is almost similar, but DMPC it is 32 angstrom. But when we calculate the just the head region as well, then it has 5, 5, 10. So altogether for POPC, it is 40 angstrom. But when it is interacting, then if you see here, sometimes the transmembrane section is extending. Sometimes it is squeezing in some cases. That is giving the a particular side. As you see from here, in some cases you can see it is going to 20 to plus 20 to minus 20. This means 40. Here we have slightly similar PC. It has it will lower overall, but it is also flexible. So whatever data we got, it is the consistent with the what we expect. So I think we are these data are okay because it is already published and it is consistent with what it should be. Yeah. We assume that definitely there is certain error, but we think that it is the maximum we can get reliability.
Okay, another question, maybe.
Did it answer your question?
Another question. I saw you run simulation up to 105 nanoseconds. What is your final simulation time you are planning?
Yeah, so that is actually a good question. It depends your facility, availability, what kind of the facility you can have and what kind of simulation you are running. So right now I am using all atom molecular dynamics simulation. This means it is very challenging. We are interested in seeing activity of each and every atom. There are also some other techniques like coarse grain. They select some of the regions as a particular particle and then they move. So here that was 105 nanosecond, but currently I have finished again around 250 nanosecond. And then our goal is to go maximum as we can. But now around 200 nanosecond. That is our intention.
Thank you. I understand that. My question was like,
Yeah.
so like this kind of biological scale, like it's like a microsecond. So
Yeah,
are
that's
you?
true. Yeah, that's also true. So what we do, uh, we want to definitely do 500 nanosecond or 1 microsecond if we can. But some of the time, when we run the simulation, for example, when we calculate the energy or root mean square deviation, if it gets flat over time, then we assume that it is saturated. Then we usually do not run. So we see how the simulation is behaving. If it is fluctuating, then we see, oh, we need to run more as well. So that is the way that we decide.
Yeah, I understand that one. Like you showed the different conformational state, but like in the sum of the simulation, like we thought that simulation is already like equilibrated, but we have to run for the longer equilibration time. So how can you justify that those are the well equilibrated conformational state of like your system?
So here I wanted to say how it behaves within that frame of time. Means right now 105 nanosecond. I'm not going to say anything what will be happening around 200 nanosecond or what will happen at 1 microsecond. That is beyond the study for this one, but that will be future what we can do. This is the way we can say. We cannot interpret its behavior exactly. That's why when I compare the conformation, then we just chose like a 16, 40 nanosecond, 80 nanosecond, or whatever we had. But these works will be keep going. If technology will get better, then people will run even 5 microsecond or something in future. In the past, people used to run around 20 nanosecond and publish the paper. So this is kind of same of thing how technology will be going on. Actually, that is very good question. I had the reviewer had the when we had submitted this paper, reviewer had this question. So I had justified from several literature and this way. So that is very good question.
Okay, thank you for everyone. Now we are going running out our time, so it is time to stop now. Okay, thank you.
Thank you so much, everyone. And if you have any further question, you can always email me, and I'll be happy to discuss and talk anything, whatever we need.
Okay, thank you to all. We are stopped here. Okay. झरिसक्यो 13जना 11जना आदर्श
अम्बा सर के छ हालखबर कुरो पनि भएछ
नमस्ते नमस्ते
रोक्नु भएन
now it's informal.
इन्द्र सर के छ तपाईसँग त काम गर्नुपर्छ तपाईसँग हामीले के गरिरहेछौँ भने यो मियोन टेक्निक युज गरेर अनि प्रोटोनको भनौँ न इलेक्ट्रोन ट्रान्सफर इन प्रोटिन भयो अनि डिएनए को फङ्सनिङ भयो त्यस्तो पढिरहेछौँ अनि सरको पनि मिल्दो जुल्दो रहेछ त्यही भएर अब काम गर्नुपर्छ सरसँग म पछि सरलाई किनभने त्यो टेम्परेचर डिपेन्डेन्ट बिहेभियर बडो क्युरियस रहेछ कि त तपाईँको स्याम्पलमा चाहिँ बाहिर तपाईँको लिपिड युज गर्नुभयो अनि वाटरको रोलहरू चाहिँ त्यति हेर्नु भएको छ कि छैन
वाटर हामीले चाहिँ अब सोलेबल साइडतिर हेर्ने हो मोस्टली हामीले अब वाटर चाहिँ त्यसमा आउटसाइड लिपिड बालेर बस्छ भन्ने एजुम्सन तर अलिअलि केले काहीँ रोल बनाउँछ अलिअलि चाहिँ डायनामिक्समा असर पार्छ त्यस्तो हामी अलिअलि हेर्छौँ
टनेलिङको काम गर्दो रहेछ त्यहाँ पनि अनि
गर्छ
ड्राई भने पनि टोटली ड्राई गर्न सकिँदो रहेछ नि त जुन सरले भन्नुभयो इभन हामी तल 4 केल्भिनमा जाँदा पनि त्यहाँ केही वाटरहरू त हुन्छ क्रिस्टलाइज त्यसले गर्दाखेरि तिनीहरू त्यो डिपेन्डेन्ट हामीले बिहेभियरबाट अचम्मको बिहेभियर देखाएको छौँ कि अहिले
त्यही त रिल्याक्सेसन इफेक्टहरू चेन्ज गर्ने त्यसैले हो कि रिल्याक्सेसन बिहेभियरहरूलाई चेन्ज गरिदिन्छ त्यसलाई हामीले अप्टिमाइज गर्नुपर्ने हुन्छ क्विकली फ्रिज गर्ने त्यस्तो हामी टेक्निक युज गर्छौँ कि नर्मली अथवा बडी चाहिँ त्यो युज गर्ने